guys, this is Holland Chambers Biology bringing to you Lecture 1 in Genetics, which deals with alleles. Now, what exactly is an allele? Well, let's take a step back and look at your DNA inside the nucleus. Now, if I were to look at just a little sequence of DNA, not the entire thing, but just a sequence of DNA, I am actually looking at a specific gene. Now, keep in mind, please stop this video at any time to write down these lecture notes, and these lecture notes should be in your notebook by the end of this unit. Okay, so sequence of DNA, for example, is ATT, CCG, GAT. Now we're going to go through transcription translation with this sequence of DNA, build a protein, and then eventually get to a trait. Well, when we're looking at a human's sequence of genes, it's a lot of information if we have to write out the entire gene DNA sequence all the time. So instead, we're going to take this DNA sequence and we're going to represent this as, say, a letter. Okay, so this specific gene, which let's say represents brown hair, I'm just going to simply call A. And so that letter that represents my gene is what I call an allele. Okay? So an allele are my letters, which are in turn my genetic genes inherited by my parents, or from my parents actually. So inherited from my parents. All right? So how did this happen in the first place? Well, let's go and look at how you were actually created. So back to basic sexual reproduction. Dad has a sperm, is going to give you half of his DNA. Mom has an egg, is going to also give you half of her DNA. And this in turn is going to create what we call a zygote, which is a fertilized egg. Okay, so this is a fertilized egg. So if Dad's DNA has a gene for big A, and Mom's DNA has a gene for little a, in your single cell, you now have big A, little a. And so you will have brown hair. Now keep in mind that these gametes, which are your egg and your sperm, are haploid cells, which means that they only carry half the DNA. So Dad's got 23 chromosomes, Mom's got 23 chromosomes, and when we add those together to create a zygote, you in turn are now going to have 46 chromosomes, okay? Now, this single cell, which has 46 chromosomes, is then going to go through mitosis. And I'm sure you guys remember that. Mitosis is when we start to divide. Divide in half again, divide in half again, until eventually we get this cluster of cells. Now, this cluster of cells is actually known as a blastula. And this is a more simplified version of what it really looks like. But long story short, you get half your genetics from your mom and dad, creates a cell, which is known as zygote. That cell then just gets stimulated to start to divide. And so all of these single cells, the blastula, every single one of these has 46 chromosomes. And they're all exactly like each other. Okay? Eventually, this blastula is going to start to differentiate, which means that a section of cells is going to start to make your eye. And then another section of the cells is going to start to make your ear. And then another section of the cells is going to start to make your heart. And I believe it's the neuron is one of the first things, specialized cells, that the blastula actually ends up making. Why? Because your body is connected by millions of neurons to help communication. Okay? So these cells are now differentiated. And once they are differentiated, they can no longer make other cells. So they can only make ear cells. They can only make heart cells. Okay? So these are specialized cells. Once we differentiate our cells, we then continue to grow, and eventually we create our organism, or our baby. Okay? Now, within our baby, we're now going to need to read what all of these different genetics are. And so let's say, for example, Dad gave the baby a big C, Mom gave the baby a little c. Another trait, so this will be trait 1, another trait, let's say, is big B, little b. Maybe that's for brown hair. Maybe this is for curly hair. And let's say big F, little f represents a third trait, which, I don't know, represents freckles, okay? So the baby now has all of these genetics, which is known as the genotype, okay? So this is the baby's genetics or DNA sequence of genes. So these are the genes, okay? Now, this genotype in turn is going to give rise to what the baby physically looks like. So physical traits, which we call in genetics the phenotype. Okay? Now the phenotype is either going to be a dominant trait or a recessive trait. So if it is expressed, we represent that as dominant. And if it's a trait that is hidden, we reference that as a recessive trait, okay? Now, what do I mean by that? Well, if it's expressed, all you need is one capital letter. That's it. So the dominant can be big A, or it can be big A, little a. Okay, as long as you have the one capital letter, that physical trait will be dominant. Say like brown hair. Hidden means that you have a recessive trait, so a lowercase allele, and you can only see it if you inherit two of them. Okay, so this one, the recessive, might be, for example, blonde hair. All right, now how did we come up with all of this? Well, these letters, aka your alleles, are organized according to being homozygous or heterozygous. Okay, so homozygous, homo in Latin means the same. So that means that the letters are the same. So big A, or little a, little a. And so again, if it has a dominant allele, this particular baby will have brown hair. If it has homozygous recessive, this baby will have blonde hair. Hetero in Latin, on the other hand, means different. So hetero means different in Latin, and therefore the alleles are different, big and little. But because they inherited a big letter, a capital, that means that that trait will be expressed and that baby will have a dominant trait. All right? So for example, let's just say big A, little a would be brown hair, and then little a would represent blonde hair. Okay? So, one more time. When we are dealing with genetics, okay, we're going to be representing a sequence of DNA as a single letter. So instead of saying the sequence, we're just going to say this whole sequence is letter A. Now this letter, we're going to inherit one letter from mom and one letter from dad, and then that in turn is going to create us, which is a diploid cell. So we have 46 chromosomes. That diploid cell is going to go through mitosis. It's going to divide to create a blastula, which will be important when we get to genetic engineering. That blastula will start to differentiate into groups of cells that can only make that specific trait. And that in turn is going to help us to create the organism. Now the baby's genetic gene combination is known as a genotype. So these are our genes. And that genotype is going to give rise to our phenotype depending on what is dominant and what is recessive. Okay? So let's go ahead and try to do an example of a genotype problem. And these problems, if I'm looking at just one trait, is known as a monohybrid. So mono means one. And again, my one trait that I'm looking at today is hair color. And so whenever you guys do a monohybrid, you always need to make sure to write out your key. And this will be really, really important when we get into dihybrids, which are two trait crosses. So the genotype with our specific example is big B. I'm going to say represents brown hair. And again, notice that I'm switching letters. I can use A, I can use B, I can use C. Doesn't matter, okay? But in this particular problem, big B is going to represent brown hair, which again, all I need is one letter. So I can be big B to have brown hair, or I can be big B, little b, and I will still have brown hair. If I inherit a little b, that means that I will have blonde hair. but in order to get that recessive trait, I must inherit 2 little b's. Otherwise, I will have brown hair, okay? So let's say, for example, you have 2 mice. Here's Mama Mouse, their big ears. She is heterozygous for brown hair, okay? And then you have Daddy Mouse over here, who is also heterozygous for brown hair, so big B, little b. My question to you guys is, what will their kids look like? So in order to figure this out, I'm sure you guys have seen this before, we're going to be using what we call a Punnett square or a monohybrid cross. So you just make 4 squares, and we're going to go ahead and put those gametes on the top and the side. So let's put Dad's gametes here. So Dad's going to either put a big B in a sperm or a little b in a sperm, okay, because he can only give half of his DNA. Mom, so this is Dad, Mom is going to put half of her genetics, big B, in one egg, and then little b in the other egg. So this is Mom, and again, this would be the sperm and the eggs. So when this egg connects with this sperm, the zygote, or the baby that we can create, is going to be big B, okay? When this sperm connects with this egg, we now end up creating a heterozygous child. Now, please remember with genetics to always write the capital letter first so we can see very clearly that this specific organism has a dominant trait, okay? So do the same thing for here, drop it down, bring it over, so big B, and then little b, all right? And again, these are your zygotes that will eventually create your organism. Now, this is not your actual, okay? This is your probability. So this is when these parents mate, you have a 1 in 4 chance of having a child that will be blonde, okay? That's not every time, it's just 1 in 4, all right? Now, this process of separating out our gametes, this is known as the law of segregation. And we'll get into that a little bit more when we talk about Mendel. But pretty much what it says is that we're just going to split the DNA in half, so that way our species stays consistent with 46 and 46 chromosomes, okay? Now, with genetics, what's going to be new, which is what Mendel brought into the game here, is statistical analysis. So when we statistically analyze our probability, you need to write out genotype and phenotype, because these aren't the actual. These are just the probability or the expected outcome, okay? So the first baby can be genetically big B. Well, that's all I have is 1. So I'm going to cross them off, write 1, ... So I'm creating a ratio. The next baby could be big B, little b. But I actually have 2 chances of that happening with this specific set of parents. The last baby, I can have a genetic disposition of little b, cross them off, okay? So now that we're done with our Punnett square, all you need to look at now is the genetics. So genetically, if I have a big B, that means physically I will have brown hair. And so 1 baby here, 2 babies here, equals I will have 3 mice with brown hair. This next baby over here has little b, which is completely recessive, which means that they will have blonde hair. And how many do I have? I have 1, okay? So this is your answer to your problems. So today when you guys go to do your monohybrid crosses, please make sure to not only write out the answer, so you need to show your work, but you also need to include a key and also the genotype, phenotype, and put a box around your genotype, phenotype, so we can actually correct it in class more efficiently. Okay, let me know if you have any questions. Otherwise, this is Holland Chambers checking out.